So welcome to Biomindscape. Today we are going to know about the mTOR, that is mechanistic target of rapamycin, a serine threonine kinase that control the cell growth, proliferation, metabolism and survival. It works by the two complexes, the mTOR C1 that is rapamycin sensitive and mTOR C2 which is rapamycin insensitive. The mTOR C1 is rapamycin sensitive. It controls the cell growth and metabolism. The mTOR C rapamycin insensitive control the cytoskeleton and cell survival. So the core component of mTORC1 pathway is insulin. The role is to activate the pathway. Then PI3K, that is the phosphoinositidil 3 kinase which produce the PIP3. The role is to produce the PIP3. And the molecule AKT, that is a protein kinase B which activate the cell survival signal. Then TSC1 and TSC2 complex inhibit the RAB role. So the RAB GTP is a molecule and its role is to directly activate the mTOR C1. Then mTOR C1 its role is the central kinase that promote the anabolic metabolism. Then S6K1 and 4E BP1 the role is to downstream the target to promote the protein synthesis. So mTOR C1 activation pathway, we will know about it. So first is the growth factor, example insulin that binds to its receptor. The receptor, insulin receptor or IGF-1R. This activate the PI3K, that is the phosphoinositidil 3 kinase. Then the activation is done. PI3K kinase, it getting converted into PIP2 to PIP3, that is PIP3, which is membrane lipid that recruits the AKT, that is PKB, to the plasma membrane. So AKT gets activated. This activated AKT is fully active by phosphorylation, that is attaching the phosphate group, and it includes the mTORC. The active AKT phosphorylates TSC2 that inhibit the TSC1 upon TSC2 complexes. The inhibition of TSC complex will further activate the RHEB, and TSC1 and TSC2 normally inhibit RHEB by converting it to the GTP, Rep GTP to Rep GDP, and when TSC2 is inhibited, this RHEB remains inactive. This Rep GTP form and this Rep GTP directly activates mTOR C1. Now this mTOR C1 is now on. Then mTOR phosphorylates. Then this particular S6K1 activates the ribosomal biogenesis and translation. Then 4E BP1 is further released, and it enhances the cap dependent translation, which results in increased protein synthesis, lipid synthesis and nucleotide synthesis, and it inhibits the autophagy. So mTOR C1 inhibition pathway, when energy or nutrition are low, at that time AMPK, that is the AMP activated kinase, is activated under low energy, which increases the AMP. So AMPK activates TSC2 and it inhibits the REB, and which further inhibit the TOR C1. So the outcome is it decreases the protein synthesis and increases the autophagy, that is recycling of the cell component. Then nutrient sensing happens, especially the amino acid especially leucine, arginine also activate mTOR C1. They signal via the RAG GTPase, which help in recruiting the mTOR C1 to lysosome. So at lysosome, Rep GTP can activate mTOR C1. So mTOR C2 is not a part of the nutrient sensing pathway. It activated by the growth factor. It activates AKT, the serine 473 site, and which is fully activated and regulate the cytoskeleton, cell survival and ion transport. So mTOR C1 has 5 components. The mTOR, which is the catalytic subunit of the complex. Its regulatory associated protein of mTOR is raptor, the mammalian lethal with Sec13 protein 8, that is MLST8, also known as G beta L, and proline rich AKT substrate 40 kilodalton, and DEP is a domain which contains the mTOR interacting protein. It is represented by ToR. So raptor, ToR, and these are the names of the 5 main components of mTOR, which are the catalytic subunit of the complex. The mTOR. What is mTOR? It is a mechanism target of rapamycin, a master controller that decides whether a cell should grow, divide or recycle. So mTOR complex. There are 2 forms of mTOR complex in the cell, that is the mTOR C1, which build, and the sensitivity to the rapamycin that control the protein synthesis, lipid biosynthesis and inhibitions. So mTOR C2 is the protector. It is insensitive to rapamycin. It regulates the cell survival, cytoskeleton and AKT activity. So imagine your cell as a city. Everything in a city is energy use, and building material, recycling plant is carefully managed. But who's the mayor of the city? So here we have known that mTOR C1, the builder, is the mTOR C1 and the protector is the mTOR C2. So both of them regulates the cell activities, cell survival. They are important in the cell growth, division and recycling. They decide whether the cell should grow, divide or recycle. So first we will focus on the mTOR C1, that one that decide whether the cell has enough resources to grow. So what activates the mTOR C1? To activate the mTOR C1, the cell needs 3 types of signal. The growth factor like insulin, amino acid like leucine and arginine, and energy availability like ATP levels. So let's see the active pathway of this mTOR. The growth factor triggering the cell. So imagine a insulin bind to a receptor on a cell membrane. This activates the PI3K, an enzyme that convert the PIP2 to PIP3 in a membrane. Now what is this PIP3? That recruits the AKT, also known as protein kinase B, to the membrane, where it activates the PDK1 and mTOR C2, that is second mTOR. So there is TSC complex and Rep complex. So AKT now phosphorylates, that is adding a phosphate group, and inhibiting means it doesn't allow inhibiting a complex which inhibit, that doesn't allow the TSC1 and TSC2, a natural breaker of mTOR C1. So TSC2 blocks a small GTPase called RHEB, but when TSC is off, this RHEB become active because there is no inhibition to that particular RHEB. So in this the GTP bound is formed. So activating the Rep GTP directly turns on the mTOR C1, but only if the amino acid are available. So amino acid are sensed by special protein called the RAG GTPase. These RAG protein help in the binding mTOR C1 to the lysosomal membrane where the Rep is waiting. So when mTOR C1 is at the lysosome and Rep1 is activated, the mTOR is fully activated. So what does the mTOR C1 do? The activity of mTOR C1 act as a constructing site. That means they are building up the cell. So stimulated ribosomal biogenesis. What does it phosphorylate? The stimulates ribosomal biogenesis. Then these are the names of the of the component which are phosphorylated, then which results the cells start to grow and more protein and more membrane and more energy is being generated. So what if the resources are low, when the nutrition or energy are less? So at that time the protein called AMPK gets activated. The AMPK sends high AMP upon ATP ratio, a sign of a lower energy. This AMPK activates TSC2 and blocks the REP. So inhibits the raptor, which slow down the mTOR C1. So what is the outcome of it? The growth is stopped and autophagy start. So cell recycle and part to survive. Now let's know about the mTOR C2, the other side. So while mTOR C1 is building, mTOR 2 is protecting. That is, it activated by the growth factor. It phosphorylates AKT at serine 473, which is fully activation. It is controlled by the cytoskeleton by PKC and ion balance by SGK1. So the survival is an important in the survival signaling. So it has some clinical importance in that the conditions like cancer, diabetes, neurodegeneration, aging and therapy, in all these conditions mTOR have a very important role. In cancer, overactive of mTOR will divide the cell and the cancer cell are formed and uncontrolled growth is seen in the cell. And in diabetes, it affects the insulin signaling. In neurodegeneration, mTOR prolongs the defective autophagy. Then in aging, if the mTOR is reduced, then it is also related to the longer lifespan also, because the mTOR activity is reduced. Then other than that, rapamycin. In therapy, rapamycin inhibits the mTOR C1 used in transplant, rejection, cancer, aging studies. So it has some clinical importance. Then mTOR C1 is activated by the growth factor. Then it will further activate the PI3K and then it will activate AKT, which will further activate the REB and which will finally activate the mTOR C1. Then amino acid will activate RAG GTPase, which will further activate lysosomal localization. Then energy will activate AMPK blocks. Then AMPK blocks mTOR C1 while when the energy is low. Then mTOR C1 promotes protein synthesis, lipid synthesis, nucleotide synthesis, inhibits autophagy. mTOR C2 activates AKT, cell survival and cytoskeleton. So whether a cell thrives, divides or recycle, mTOR pathway is a master switch behind it all. Understanding this switch and you unlock a huge piece of biology. So the catalytic subunit of mTOR pathway, mTOR is the mammalian target of rapamycin and it is formed by two distinct protein complexes, the mTOR C1 and mTOR C2. Within these complexes, let's know the subunits or catalytic subunits of both the mTOR. So mTOR 1 is characterized by presence of mTOR regulatory associated protein of mTOR, that is the raptor. Then MLST8, that is mammalian lethal with Sec13 protein 8, and the non-core component and the deptor. These are the main components of mTOR 1. The raptor plays a important role in the recruiting the substrate to mTOR complex 1 with while the MLST8 is involved in maintaining the mTOR kinase activity, and the deptor and Pares 40, they are negative regulator of mTOR C1. Then mTOR C2 include mTOR Richter. Now rapamycin insensitive companion of mTOR, that is a Richter. So this is the main subunit of the mTOR C2. then MLST8, then M sign 1, and other components are present. The reactor is essential for mTOR2 catalytic activity, similar to the receptor role in mTORC1. Both mTORC1 and mTORC2 play important role in cellular process. So mTORC1 primarily regulate the cell growth, proliferation and metabolism, while the mTOR2 is involved in the cell survival, cytoskeletal organization and glucose metabolism. Thanks for watching.